this channel because I love teaching and learning, and I also love sleeping. I am a Bachelor of Science graduate, and I wanted to share my knowledge and love for science with the hopes of allowing you to learn while relaxing and falling asleep. We live in a culture where it is normal to always be busy and stressed. So I hope that you can take this time to wind down and learn something new. So let's get started, shall we? So today I wanted to talk about the structure of the cell. Now, there are many different types of macromolecules in your body and in plants, in many, many living creatures. And there are 4 different main macromolecules. So we have your nucleic acids, which are composed of DNA, RNA, and chromatin. And you also have your phospholipids, which are the primary component of the cell membrane. You then have your polysaccharides, which are your carbohydrates, and they're your source of stored energy. And finally, you have your proteins, which are the structural elements of the cell, and they also conduct metabolic activity. So your cell is a membrane-bound structure containing all 4 of these macromolecules, and they all work together to make sure that your cells are perfectly capable of performing many tasks to keep you alive and going and healthy and living day to day. So let's start with phospholipids. So phospholipids are a glycerol linked to a phosphate and 2 fatty acid tails. And the most common phospholipids in your cell membranes are composed of either 16 or 18 carbons in the chain. And these carbons can be linked together by either single bonds, which would make it saturated, or double bonds, which would make it unsaturated. So the phospholipids are amphipathic, which means they are both hydrophilic and hydrophobic. So the hydrophobic component is the fatty acid tails. They do not like water. But the hydrophilic part is the phosphate head, which does love water. And hence, in order for a cell to create a membrane, they want to make a bilipid membrane. And by that, it means for the hydrophilic heads to both be inside the cell and facing outside the cell. And your phosphate tails would be facing each other, as so it doesn't touch the water inside or outside, and you'll have a happy cell structure. Now, there are different types of factors that affect the fluidity of your cell membrane. So first is the number of carbons on your acid-fast you have the 18 carbons would decrease fluidity, and shorter chains where you have the 16 carbons would increase fluidity. And this is because when you have longer chains, you have them packed together more tightly than shorter chains. Therefore, when you're packed tightly, it decreases fluidity. And when you're not packed as tightly, it increases fluidity. So you just have to remember, longer chains decrease fluidity, shorter chains increase fluidity. And the second type of a factor is the bonds that hold the carbons together. So we discussed before that when you have single bonds, you would have a saturated fatty acid. But when you have double bonds between the carbons, you would have an unsaturated fatty acid. And the double bonds cause kinks in the chain. So it causes a bend in the fatty acid, which is why they are unsaturated. It pushes the neighboring phospholipids because, of course, they are bound together to ensure the cell has a full structure. So they're all congregated together. And when you have double bonds and there's a kink in it, it kind of pushes the neighboring phospholipid and says, hey, give me some space, right? So now you have a more fluid membrane because it's not packed as tight as a saturated phospholipid would be. Another factor, which would be number 3, would be temperature. So temperature increases fluidity when it's at high levels, and lower temperatures decrease fluidity. And this is because when you have higher temperatures, they expand the cell, and when you have colder temperatures, it compresses and becomes more stiff, so it decreases fluidity. You can kind of think of it as when you yourself are in very high temperatures, you kind of want to just splay out and make sure every limb is not touching each other. But when you're cold, you want to hug yourself and you become compressed and stiff. Okay, so the final factor here is the presence of steroids, such as cholesterol, in the lipid membrane. So cholesterol molecules that are good to be on the lipid membrane, they flow around the bilayer. They decrease fluidity because they cause the fatty acids to pack closely to the neighboring phospholipids. So it's like thinking of a big crowd of people at a concert. If you add more people, which would be your cholesterol molecules, having more people added and added to the crowd would have less space for each person and decreases the fluidity, so decreases the movement that you can move around if you were in that crowd. But the thing about cholesterol is when it's at low temperatures, the cells begin to stiffen and decrease fluidity. So at this case, the cholesterol can actually help fluidity by keeping the phospholipids separated from each other as it moves around. So it's very variable for this factor. Just know that at high temperatures or mediocre temperatures, it packs closely and decreases fluidity. But low temperatures only, it may actually help increase the fluidity. So that's all I have for this video today. I wanted to keep it short and sweet since this is my first video and I wanted to test the waters a little bit. I will be doing many more segments of each topic and perhaps compiling them into one big long video when they're all finished, so you can have a relaxing sleep and just be able to listen to a long video learning about science and specifically biology for this segment. Thank you so much for joining today. I hope you've learned something